Bonjour à toutes et à tous. Welcome, everybody. My name is Viktor Senderov. And I'm going to talk to you about the probabilistic programming approach to investigate the coevolution of genes and phenotypes in birds. I'm a computer scientist currently with Hélène Morland's lab in École Normale Supérieure in Paris. Previously, I was with Fredrik Ronquist at the Natural History Museum in Stockholm, Sweden. So what I'm interested in is using probabilistic programming languages to elucidate the phylogenomic basis of bird color pattern formation. And to whet your appetite a little bit, here on the picture you see some of the extraordinary colors that the estrudid family of finches display. In this talk, I'll talk about three things. First, I'll talk about bird coloration, what it is, what exactly are we trying to do. Then I'll present our mathematical model, and then I'll conclude with this remark on probabilistic programming. So we have to be precise when we talk about what exactly we're doing. Bird coloration has been studied in the sense of what are the evolutionary drivers. It's pretty well understood. It could be signaling, it could be predator avoidance, camouflage, and whatnot. What is known as well, pretty well, is how overall bird colors form. They could be through pigments or through structures that interfere with light. But we know why they appear and the developmental mechanisms. For example, here you have these rock pocket mice, which have a darker morph on lava substrates. And the reason is for camouflage, and the mechanism with which it appears, it's a mutation in a gene called melanocortin 1. But what is less well understood is how bird color patterns evolve. And I recently thought of a good analogy to explain what bird color patterns is to think of them as children's coloring books. So essentially, those are the various domains, their borders, and some special features such as stripes, dots that may form. And then you kind of fill in the domains with a specific color, and the idea is that related species will share kind of a similar pattern, even though their colors might be slightly different. And so we don't know like which genes drive that and how they form developmentally. So we want to study this. So our colleagues from the Marceau lab at Collège de France did like a small scale study where they looked at these finches. Here the zebra finch, the Gullian finch, and they developed a method to characterize these color patterns. And then we want to scale this up. So we selected 350 bird species from across the tree of birds, for which we have genomic data. And then we went to the Museum of Natural History and we photographed these 350 species. And some poor student will take a ruler and measure certain measurements, and then they will form kind of this phenotypic data of the type that is shown here on the slide. And we have another student who needs to retrieve the versions of those genes that have been selected from our literature studies, which are our candidate genes. So listening to the previous talk by Sina, and also what Kirti was saying today, maybe we should do something better than blast search, but this is what our student did so far. We're not quite ready with the data set, so we might still do something like prot-BERT or something like this. But the idea is that we want to get the versions of these genes for our bird species. So to wrap up, our data set is a phylogenetic tree. And the tips of that tree are enriched with this genomics data and the phenotypic data. And then we want to test which genes from these candidate genes, or if any are kind of related to these patterns, if they're responsible somehow. And I want to give a brief overview of methods which exist already to do this. GWAS doesn't work in this case because GWAS is a regression, and in kind of like the most general way you do it, you assume the independence of the samples, so they have to be IID samples. You can do something that's called phylogenetic generalized least squares, which is again like a regression-based method which does control for the non-independence of the samples. And why are our samples not independent? Well, it's exactly the phylogenetic structure which gives the degree of relatedness of the samples. But if you just do this kind of like a regression-based thing, you are not you're not incorporating the evolutionary process. So there are several methods that do incorporate the evolutionary process that have been proposed in the literature for this type of things. Some of them are based on this ratio of non-synonymous to synonymous mutations, or DNDS ratio. So essentially you split your data set in two parts, one that has the phenotype and one that doesn't, and you look for genes that have high DNDS ratio in your phenotype, but not in the other part. The problem is that the NDS doesn't always means positive selection. It could mean negative selection, or in certain special cases, even neutral selection. So there's another type of methods which try to overcome this, called profile methods. Without going into too much detail, they introduce like an amino acid profile, which tries to take directionality of selection into account. But as far as I'm aware, at least the ones that I checked, they go site by site. So they give a degree association between a site, and a site could be a nucleotide, a codon, or an amino acid, and some phenotype. But what we wanted to do is we wanted to associate the whole gene with the phenotype of interest. So our method is based on somehow inferring evolutionary rates, but it's a revolution in phylogenomics, inferring evolutionary rates. And let's talk about our methods. So the cute birds method, and I realized that pasting from Inkscape into PowerPoint gives some quirks here. The cute birds method is based on in inferring these three rates. So think about it this way. So you have some phenotype, in this case, what a striped phenotype, and then you have a sequence, and then you have a rate at which they evolve together. So think of this as a Poisson process. So whenever a jump occurs of this type, then both the sequence and the phenotype change. And we have these base rates where this is kind of the molecular substitution rate and the phenotype transition rate. And the idea is that if the gene and the phenotype are linked, then this rate will be more than 0. And if they're not linked, it will be small. And we want to do this in a Bayesian setting. And I always introduce a slide with the Bayes rule in my talks. And the reason why I'm introducing it here is because we're also doing it in a probabilistic programming setting. And later on, it will become clear. But the way to specify the likelihood is by an observe or a factor statement in a probabilistic program. But what's the mathematical formulation? The mathematical formulation is that we use this message passing schema that is here on the right, and we actually do a technique called data augmentation, which means we explicitly sample these jumps from this rate, the new rate, and then we enrich the tree with these jumps, and then we do some clever continuous and discrete time Markov chain stuff to compute the messages, which then lead to the computation of the tree likelihood. And you could So here you had these matrices here. So you could parameterize the matrices so that it's not just like the classical symmetric Jukes Cantor. You can actually have something cleverer, such as the Kimura model, which is a better approximation of the evolutionary process. We do have some limitations. For example, the rate should be the same for all sites of the gene, but not necessarily the same for all genes. Right, and why did we need this data augmentation? I'm not going to spend too much time, but unless we do it, this matrix will blow up. Here I'm giving like just like a small sample for a gene of length 1, but if the gene is, let's say, of length 100, then the dimension of the matrix would be something like 2 to the power of 201. Right. So far, as I said, we're still gathering the data, exploiting poor students, and yeah, I should really thank my collaborators again. But I only did simulation studies, but simulation studies are promising. For example, if you simulate data under the kind of independent evolution, so you let a gene evolve and you let the phenotype evolve with certain rates, but you keep this coevolution rate to 0, then you get a pattern like this. And if instead you increase the I don't want to say coevolution, joint evolution rate, then you get a pattern like that on the right. And the ideas here a little bit think of like this marginal distribution tables and joint distribution tables in your basic probability textbook. Even though you might not see any difference in if you marginalize out. I mean, here we have the same amount of A and the same amount of A and the same amount of the green phenotype and the same amount of the red phenotype, but they co-occur in different proportions because of this correlated process. And then if I use the simulated data and I infer back the rates, you do see this, you do recover this elevated joint rate, which would be a signal of the correlated evolution. Yeah, so far about our method and results, and I promise I'm going to talk a little bit about probabilistic programming because we're doing all of this in this probabilistic programming framework. So let's take a step back and think about why do we model stuff. And the reason why we model stuff is because the system that we want to study is usually intractable to kind of study directly. Like it's too big, too expensive, doesn't exist anymore if it's evolution. So you want like an abstract representation of your system, and then you do your analysis, your reasoning, your design on the abstract representation, kind of as a proxy for the real thing. And my claim here is, I'm not going to prove it, I'm just going to state it, is that universal probabilistic programming languages give like a holistic solution for your modeling needs. At least in the case when the modeling needs are kind of statistical modeling type of modeling. If you can do like a Bayesian model of your system, then with the probabilistic programming language, you can express that in a very succinct way and solve it in a very nice way. So let's see an example. This is not a phylogenetic example because those models are a bit more complicated, but it's kind of the hello world of probabilistic programming languages. Sometimes it even appears in these like coding interviews. If you go via quant or something like this, they might ask you something, you can solve it in like 5 lines of probabilistic programming code. Essentially a probabilistic programming language, syntactically, whoops, do I have a transition here? Yes, I think I do. I don't know how to stop this. I'll just go to this slide. Syntactically has these keywords: sample, which means to sample from a probability distribution, and observe, which means to condition your computation, the likelihood of your computation on some data. And also they have this higher order construct called infer, which I didn't want to discuss too much, but after seeing Emilia's talk, I'm probably gonna discuss it a little bit more. The infer construct simply tells the compiler which inference to use, and the probabilistic programming languages give us this flexibility not to have to implement the inference. So I can plug in here SMC in the case for sequential Monte Carlo, or MCMC in the case for Markov chain Monte Carlo, or whatnot. It doesn't really make any difference for the program that I write. This is kind of like they have a true separation of concerns between inference and modeling, right? So this is the syntax, and now it's going to shift. It doesn't shift now. I don't know what happens. And maybe I talk just a little bit about semantics. And sorry for the computer scientists here, this is not neither the operational nor the denotational semantics of probabilistic programs. It's just a bit of an intuition about what probabilistic programming languages mean. So a regular program, it just goes from input to output, and it has some side effects. Probabilistic programs, I mean, it doesn't really help so much, but if you think of a single execution of a probabilistic program, it's like you have some hyperparameters, some data, and you get some sample from the prior, essentially, and the score, like this likelihood. But if you combine this with the inference, and if you look at the distribution over traces, then essentially you get the posterior distribution in the Bayesian sense. And in this case, the posterior distribution happens to be this, so we can say this coin is probably fair, even though who knows from where it came with these like crazy aliens. It came from Bing, right? So we have our own probabilistic programming language for phylogenetics called Tree People. We're going to release it in October, and this is a bonus slide which I'm not going to talk about now, but I could if there are questions. And thank you all so much for your attention.
Thank you, Viktor. Do we have a question?
No, just say what you were about to say.
The question is what you wanted to do. What did you want to say?
I don't know what I did. What did I press? So on this slide, this is just the question would have been, what's the architecture of Tree People? How did we construct it? And it's based on this miking system, which is bootstrapping, but the initial miking compiler is written in OCaml. And then the thing that for you is interesting here is that there's a pluggable architecture where you can put all these inference strategies, right? So right now we have lightweight importance sampling, sequential Monte Carlo with a bootstrap particle filter, a live particle filter, lightweight MCMC, trace MCMC, particle MCMC. But you could write another one fairly easy and extend the compiler with that. And also we have a bunch of static transformations in the program, such as conjugacy analysis, suspension analysis, and whatnot. So these are all, the idea is that the compiler is very modular, and you can add stuff to make your inference better in a fairly easy way without changing the model, without touching it. It's all kind of separated away in the compiler.